What is the fundamental unit of everything that breathes, grows, or reproduces? Welcome back, everyone. Today we are breaking down the absolute foundation of IB biology, cell structure and microscopy. Whether you're trying to master magnification math or unlock the HL secrets of endosymbiosis, we are covering it all. Let's dive in. Here is our game plan for today. We'll start with the fundamentals of cell theory and then microscopy skills you must know for paper 3. Then we'll map out our cellular architecture across prokaryotes, eukaryotes, and the weird atypical cells that break the rules. Finally, the HL students will explore how complex life actually evolved through endosymbiosis. And let's kick off things off with cell theory, which rests on three core pillars. All living organisms are made of one or more cells. Cells are the smallest, most basic unit of life, and all cells come from pre existing cells. The IB expects you to use deductive reasoning here, meaning, if we discover a brand new bizarre organism deep in the ocean tomorrow, we can confidently predict without even looking that it will be composed of cells and rely on cellular division to reproduce. Moving on to microscopy skills, for your practical exams, you need to know how to prep a temporary wet mount. You place your specimen on a slide, add a drop of water or a stain like methylene blue for animal cheeks or iodine for plant starch, and lower the cover slip at a 45-degree angle to prevent nasty air bubbles. When focusing, always start on the lowest power lens. Use coarse focus knob first and only switch to fine focus knob when you dial up to high power, which is 0.5 m is equals to I upon A. Now, for the math that terrifies everyone is actually incredibly simple. Magnification equals image size divided by the actual size, as told before, which is m is equals to I upon A. Remember, image size is what you measure on paper with your ruler, and actual size is the real microscopic size of the cell. Crucial tip: always convert your ruler measurements into micrometers before doing the math. But remember, 1 millimeter equals 1000 micrometers. So if your image is 40 micrometers, that's 40,000 if your image is 40 millimeters, that's 40,000 micrometers. To calibrate this in the lab, you will use an eyepiece graticule, the tiny ruler built into the lens, and match its divisions against a own stage micrometer scale on the slide. Technology drives science, and light microscopes are great, but electron microscopy (EM) completely changed the game by offering vastly higher resolution. Because electron wavelengths are much shorter than light waves, techniques like freeze fracture have allowed us to snap cells frozen have uh, techniques like freeze fracture allow us to snap cells frozen to split open lipid membranes and see inside them. Cryogenic electron microscopy let us view proteins in their natural frozen state without altering them, while fluorescent stains and immunofluorescence use glowing antibodies to tag specific target proteins in real time. Despite the massive diversity of life, every single living cell on Earth shares exactly four universals: a lipid plasma membrane to control what enters and exits, a fluid cytoplasm where metabolic reactions happen, uh, DNA as the genetic blueprint, and ribosomes to synthesize proteins. No matter how weird a cell gets, it has these four. Let's look at the simplest architectural design, prokaryotes, which are your bacteria. They are non-compartmentalized, meaning they don't have no internal membrane-bound organelles. Instead of a nucleus, they have a nucleoid region containing uh, naked circular DNA. They have smaller 70S ribosomes, a rigid cell wall, tiny extra loops of DNA called plasmids, hair like villi for latching onto surfaces, and a whip-like flagella for swimming around. In contrast, eukaryotes are all about compartmentalization. Think of them like a mansion with specialized rooms or organelles, separated by membranes, allowing different chemical environments to exist simultaneously. We are talking about a distinct nucleus holding linear DNA, a rough ER studded with larger ATS ribosomes for protein transport, a a smooth ER for lipid synthesis, a Golgi apparatus to package those proteins, mitochondria to generate ATP, plus lysosomes, vacuoles, vesicles, and a structural cytoskeleton framework. Summarizing the major architectural differences for your notes, prokaryotes have naked circular DNA in a nucleoid, smaller 70s ribosomes, and no internal compartments, typically measuring small to at 1 to 5 micrometers. Eukaryotes have protein bound linear DNA inside a nucleus, largest ATS ribosomes, and distinct membrane bound compartments, typically measuring much larger at 10 to 100 micrometers. Even if you are just a single cell, you still have to accomplish the seven functions of life: metabolism, response, homeostasis, growth, reproduction, excretion, and nutrition, which can be learned in an abbreviation called Mr. H. Grill. Take paramecium for example. It eats food particles via an oral groove for nutrition and expels water using a contractile vacuole for homeostasis. Chlamydom, uh, Chlamydomonas, a single-celled alga, uses an eye spot to swim toward light for environmental response and creates its own food via photosynthesis for nutrition. Moving to multicellular eukaryotes, we see major structural variations. Plants have rigid cell walls made of cellulose, contain chloroplast, and feature a massive central vacuole. Fungi also have cell walls, but they are made of chitin, and they never have chloroplast. While animals completely lack a cell wall, allowing them to change shape, and they only use small temporary vesicles instead of large vacuoles. But biology loves exceptions, and there are atypical cell structures that challenge standard cell theory. Skeletal muscle fibers are incredibly long and contain hundreds of nuclei working together in one continuous cytoplasm. Mature red blood cells throw away the nucleus entirely to make room for oxygen. And similarly, phloem sieve tubes elements in plants hollow themselves out, losing the nucleus and most cytoplasm to allow sap to flow cleanly through the stem. The IB loves testing your ability to read real micrographs, so don't look for perfect textbook drawings, look for patterns. A large dark circle is almost always the nucleus. Ovals filled with wavy parallel internal lines are the mitochondria, and parallel sheets or lines packed tightly toward, together near the nucleus represent your rough endoplasmic reticulum. When drawing these yourselves on an exam paper, follow the strict IB layout rules. Use sharp, continuous single lines with no sketchy artistic shading, and ensure your labeling lines are perfectly straight, drawn with a ruler, and point directly to the structure without crossing over one another. That wraps up our standard level core. Standard level students, feel free to stick around to look ahead, but for my higher level students, let's step up the complexity and look at how these compartments evolved in the first place. According to the endosymbiotic theory, billions of years ago, large primitive cell engulfed a smaller free living prokaryote via endocytosis. Instead of digesting it, they formed a symbiotic relationship where the small cell got protection and the large cell got a massive energy boost. This is exactly how the mitochondria and chloroplasts evolved. The proof is written right in their anatomy. Both mitochondria and chloroplasts have their own 70s ribosomes, their own naked circular DNA, a double membrane from the engulfing process, and they replicate independently via binary fission, just like modern bacteria. Next up is cell differentiation. Every single somatic cell in your body contains the exact same identical DNA code. So how does a skin cell look and act completely different than a brain cell? It all comes down to gene expression. During development, specific chemical signals switch certain genes on while permanently turning others off. And by expressing only specific subset of genes, cells differentiate into highly specialized structure. Finally, the evolution of multicellularity answers why organisms bother moving beyond a single cell. Going multicellular offered massive uh, evolutionary advantages. It allowed organisms to grow larger without being restricted by surface area to volume limits, live longer if individual cells died, and form complex cooperative cell networks where different tissues could specialize in specific tasks. And this is a complete wrap on the topic. We covered the rules of cell theory, how to handle the magnification triangle, the structural differences that separate prokaryotes from eukaryotes, and the evolutionary history of multicellular life. Make sure to download the summary notes in the description below. Drop a like if this helped you study, and I'll see you all in the next video.